Hello viewers, we were discussing about enzymes chapter. In that chapter, we are going to discuss a topic called as enzyme inhibition. What is enzyme inhibition and what are the types of enzyme inhibition we are going to discuss in this session. Let us begin with the definition of enzyme inhibition. Inhibit means stop or decrease or stop. Hence, the action of enzyme activity is going to be reduced or stopped by certain compounds, and the process is called as enzyme inhibition, and the substance which involved in this inhibition process is called as inhibitors. So let us study this topic. A wide variety of compounds, they may be organic or inorganic, can reduce enzyme activities, and these compounds are called enzyme inhibitors. So the compounds which may be organic or inorganic, they are going to bind to the enzyme and thereby inhibit or stop their activity. Enzyme inhibitors binds to the enzyme and reduce their activity. This process is called as enzyme inhibition, and the compounds which are involved in this enzyme activity reduction process is called as enzyme inhibitors. So let us discuss what are the types of enzyme inhibitions. There are different types of enzyme inhibitions. Among them are one competitive enzyme inhibition, non-competitive enzyme inhibition, and uncompetitive enzyme inhibition. These are the three enzyme inhibitions, and if you go through their names meaning, competitive means there is a competition between the inhibitor and the substrate for the enzyme, because enzyme is going to bind to substrate. So the substrate will have a competition for the enzyme, and inhibitor is also having competition for binding to the enzyme. So there is a substrate and inhibitor competition for the enzyme. That's why it's called as competitive enzyme inhibition. Non-competitive, where there is no competition between the substrate and inhibitor for the enzyme. And uncompetitive means here you can see the different action of inhibitor which are going to bind to enzyme substrate complexes. So let us discuss these inhibitions in the detail. Competitive enzyme inhibition, as the name itself indicates, competitive enzyme inhibition. In competitive enzyme inhibition, the inhibitor is structurally similar. The inhibitor is structurally similar to that of the substrate, and hence it is a substrate analog. Hence inhibitor competes with the substrate for binding with the active site of the enzyme. This paragraph 5 we are going to see in detail along with the diagram. If you see in the diagram the enzyme, and if you see the shape of the substrate above, which is in white color, triangular shape, and if you see the inhibitor below, which is also triangular in shape, but it is in black color. If you consider the shape of this substrate and inhibitor, both are having the same shape, and hence it is written in the first line of the paragraph, the inhibitor is structurally similar to that of the substrate. So inhibitor is similar to that of substrate, and the binding site of the enzyme, that is active site of the enzyme where substrate will bind, if you see the shape of the active site is similar to that of substrate, and also with the inhibitor. So if you go in the pattern of enzyme plus substrate, then you will get enzyme substrate complex and you are going to get product. That's the normal enzyme activity process. And if you see the process in the inhibitor way, you can see enzyme, and then you are if it is going to bind with the inhibitor in the active site, then there is no product, because product comes from only substrate and not from the inhibitor. So if you see the Substrate, if it will bind, it is enzyme substrate complex. If inhibitor will bind, it is enzyme inhibitor complex. And hence inhibitor competes with the substrate for binding with the active site of the enzyme. The binding of the inhibitor to the active site of the enzyme prevents the substrate from binding with the enzyme, which result in the decreased enzyme substrate formation and reduced rate of enzyme reaction, where the product will be production is less. So for example, if you see the pattern in the downwards, enzyme plus inhibitor, you will get enzyme inhibitor complex, and whenever inhibitor will bind to the active site, there is no place for the substrate to bind, hence the enzyme activity is reduced or decreased. This is about enzyme inhibition in that competitive enzyme inhibition. If you see the substrate and inhibitor, both are competing for the single binding area of enzyme, that is active site. So this process is called as competitive enzyme inhibition. Next, this in detail, taking a specific example. The formation of enzyme substrate complex or AI complex depends on the relative concentration of the substrate and the inhibitor. If enzyme is 1 and substrate is 1 and inhibitor is 1, the chances of substrate binding and inhibitor binding is 50-50, because both are one substrate and one inhibitor, and enzyme is also 1. So similarly, if you increase the substrate concentration, the substrate is 10 in number and inhibitor is in 1 in number, then the chances of substrate binding to enzyme is more, because substrates are 10 in number whereas inhibitor is only 1 in number. So the this type of inhibition can be overcome or reduced. This activity of inhibitor can be reduced by increasing the concentration of the substrate. Competitive enzyme inhibition is reversible. So that means you can remove the inhibitor and make the enzyme active again. It can be overcome by a high substrate concentration. So that's what I told. High substrate concentration means if you take 10 substrates, then it can you can have a more chances of binding to the enzyme. Thus, competitive enzyme inhibition does not alter the Vmax and but increase the Km value of maximal velocity is increased. If you go through the specific examples, malonate is the competitive inhibitor of succinate dehydrogenase enzyme. If you see the enzyme in this slide, the succinate in the example second paragraph, if you see succinate dehydrogenase enzyme, that is the enzyme diagram in the box, and it oxidizes succinate to fumarate, that means substrate is the succinate, and the above if you see the uh, enzyme substrate and the product obtained, that the product is fumarate. And if you see downwards, enzyme combines with the inhibitor. For example, in this case, malonate has a structure similar to succinate. Hence, malonate competitively inhibits the succinate dehydrogenase enzyme. That means enzyme means succinate dehydrogenase enzyme. Above substrate is actually succinate. The inhibitor is malonate. Malonate which inhibits succinate dehydrogenase because it's a analog of succinate. So this is specific example. And if you see the same example continuation, succinate is the actual substrate and the malonate is the inhibitor. If you see the structure of both, you can see dicarboxylic acid structure in the succinate, and also malonate is also having dicarboxylic structure, that means two COH groups. The enzyme is going to combine either with succinate or malonate, and in case if it combines with malonate, the succinate dehydrogenase enzyme which is written below the arrow mark is going to get converted to product. Will not get converted to product, and hence it will get inhibited. Whereas if succinate will bind, you will get the product. That's why you can see in the above example, if fumarate is available, in the below there is no product because malonate is a inhibitor. In the non-competitive enzyme inhibition, as the name itself indicates, non-competition. In non-competitive enzyme inhibition, the inhibitor is not structurally similar to the substrate, hence it does not compete with the substrate for the active site. So if you see the first line in that, the inhibitor is not structurally similar to the substrate. So not structurally similar to substrate means both shape is not same, structurally they are not same. If you see the enzyme, in the upward process, if you see enzyme plus substrate, you'll get a enzyme substrate complex, and then inhibitor will bind, and then there is a formation of ESI, that means enzyme substrate inhibitor complex, and there is no product. In this case, if you see in the upward process, you can see substrate is triangular in shape and inhibitor is round circular shape with a black color, and hence they are competitive. In competitive there is a structurally they are having same, but in non-competitive the structure of the substrate and inhibitor is not same. And in the downward process, if you see, the same process of inhibition takes place, but in this the pattern of binding the inhibitor to the enzyme, it takes place in the initial stage only before binding of the substrate to form A complex. So after inhibitor binds to enzyme forming A complex, then substrate will bind. After that you will get ESI complex. There also there is no product. That means either the substrate will bind first or the inhibitor will bind first. The enzyme is going to get inhibited through ESI complex or EIS complex. That means enzyme substrate and then inhibitor, or enzyme inhibitor and then substrate binding. In both cases enzyme will get inhibited. Here you can see inhibitor is not binding to the active site, it is binding to the other than the active site. You can triangular shape is the active site, and the curve shaped area is the inhibitor binding area of the enzyme. So active site binding to substrate and inhibitor is binding other than the active site, hence it is uh, non-competitive, where there is no competition between substrate and enzyme. Non-competitive enzyme inhibition can be reversible or irreversible, but both show similar kinetics. Reversible means the enzyme will get activated to its original form by removal of inhibitor. Irreversible means it is completely damaged, because inhibitor once it is attached to the enzyme, you cannot able to remove that inhibitor from the enzyme. So reversible non-competitive enzyme inhibition. In non-competitive enzyme inhibition, there are two types: reversible and non-reversible or irreversible. Let us see reversible, which is also called as that backward process, where inhibitor will bind and then again it will get converted to enzyme plus free inhibitor. In non-competitive enzyme inhibition, the inhibitor is not structurally similar to substrate, hence it does not compete with the substrate for the active site of the enzyme. As I shown before, inhibitor may not bind to with the active site. Inhibitor will not bind to active site, it binds to curved area as shown in the figure. So here you can see the black color, it will bind to the curved area of the enzyme, which is a inhibitor binding area, and instead it may be binds to enzyme at a site other than the active site. Since substrate and inhibitor combine at different site, formation of EI and ESI complex are possible. Since enzyme substrate inhibitor may break down to form products at a slower rate than does ES, the reaction may be slowed but not stopped. That means once the inhibitor will bind to the enzyme, and even though the substrate will also bind, the product can be obtained but in a slow manner. So inhibitor does not interfere with the binding of the substrate. So inhibitor is not going to affect the binding of the substrate, but it will going to decrease the enzyme activity by reduced product formation. So inhibitor does not interfere with the binding of the substrate with enzyme, but it lowers the maximum velocity attainable. So Km value is unchanged, but Vmax is lowered. Maximum velocity is lowered, but Kmax remains the same. For a specific example, antitrypsin non-competitively inhibits trypsin in reversible manner. So that is reversible antitrypsin. And the trypsin inhibitor present in soybean and ascaris is going to affect the enzyme in non-competitive reversible form of enzyme inhibition. In irreversible non-competitive enzyme inhibition, a variety of enzymes poisons reduces the enzyme activity non-competitively. Here, inhibitor is not structurally similar to the substrate, but these inhibitors, which is reversible, this process is not readily reversible by increasing the substrate concentration. However, the presence of substrate exerts a protective role by blocking or slowing the access of inhibitor. In this process, the inhibitor, once it binds to the enzyme, that enzyme is permanently damaged. You cannot able to remove the inhibitor. And in this case, even though by adding the excess quantity of substrate, it is only protecting by blocking the enzyme, but once a inhibitor will bind to the enzyme, even the concentration of substrate increasing will not having any use, because once inhibitor will bind, that enzyme will permanently get inhibited. Examples, specific examples, for examples, heavy metals like mercury, silver is going to inhibit certain enzymes. Hence heavy metals are not good for the body because they act as various type of enzyme inhibitors, and in this case irreversible inhibition, non-competitive enzyme inhibition occurs by these type of metal ions, fluorides, cyanides, etc. Next we are going to discuss about non-competitive enzyme inhibition. And after non-competitive enzyme inhibition, uncompetitive enzyme inhibition, the inhibitor does not bind to free enzyme. It will not bind to any free enzyme, but it can bind only with enzyme substrate complex and decreases the velocity of enzyme reactions. Uncompetitive enzyme inhibition decreases both Vmax and Km of enzyme. For example, if I show you this first type of inhibition, in this competitive enzyme inhibition, you can see the enzyme substrate will bind or enzyme inhibitor will bind. That is competitive. Whereas in non-competitive, enzyme substrate and inhibitor, or enzyme inhibitor and substrate will bind, but the inhibition takes place, no product will be obtained. But in this case, it is very peculiar because here it will not bind to the enzyme directly, but it will bind to enzyme substrate complex. First you can see the enzyme and then the substrate will bind, you will get enzyme substrate complex, then the inhibitor will come and get attached to the enzyme by forming enzyme substrate inhibitor complex, and then you will not have any product, thereby enzyme is inhibited. In this process it will bind to enzyme substrate complex rather than free enzyme. This is the difference between non-competitive and uncompetitive enzyme inhibition. Specific example, inhibition of placental alkaline phosphatase enzyme by phenylalanine, inhibition of S-adenosine methionine transferase by ATP. These are the specific examples for uncompetitive enzyme inhibition. Remaining topics we'll discuss in the next session. Thank you.